On that note, another one of these guys ruins the shot. I'm kind of done with you, guy. Go somewhere else. Play.
And our depth is 408 meters.
Yeah, so we've gone 100 meters vertically. And what are we at, 300 or 400 meters distance?
I'm not
sure. Do
sure.
you know the last?
200 plus 250. 450. 450 meters. Yeah.
It's pretty good. 450 meters we've traveled.
Wow. Guess we've been focused so much on the feeding habits of the benthos here. I missed the call.
We're just hopping along looking for more of these bacterial mats, and we've been very successful finding them for a long time. Though when we were closest to the actual targets of the plumes was where we found the largest, very extensive bacterial mat.
See, find out.
And bubbles.
And bubbles.
Yeah, we haven't seen a lot of bubbles, a lot of locations for bubbles, but we did see quite a bit of bubbling going on in one of the locations.
Yeah, a little
where the mats were extensive, as Cheryl was saying.
And this is the very tiny bacterial mat. And if you can see those little tiny white tubes in there, someone, I don't know if it was yesterday or the day before, was suggesting that that might actually be made by bacteria also.
Bulldozing a little bit. Okay.
Move complete. Thanks, Bridge.
We're still going upslope here.
Yeah, feel free to change your heading. Understood.
I don't know what's on the left there. There's a couple mats here to keep our eye on, but I'm going to peek at whatever this
looks like
fish on the
a
bottom
different
or something
fish
is.
than we've seen previously. Or at least today.
A little bit more activity in terms of biomats here, so that's good. Pick that back up. Okay, video, you can start it on the fish.
Hopefully it will stay still for us just for a moment. Yeah, different coloration.
Wow.
Settling down.
Kind of has the color of that green eye, but
I was going to say, but it doesn't have the eye.
right.
It has everything else.
It's a beauty. Very pretty fish.
There we go.
Goes some squid.
Couple of biomat spots here.
Yeah.
A few off to the side there.
Are those the suggested shrimp up in the water column? On camera too.
Oh yeah. I can only hope.
Yeah, so a few dives ago when we saw the whole bottom of the sedimented slope covered with brittle
Yeah, go
stars,
ahead if you want it.
and we were wondering how
Didn't
many
seem
there
like
would
there's
be if
anything
you were
going
able
on.
to sweep them all up into a huge pile and count them. And today we can wonder that about these quill worms.
Yeah.
There's a little hole in this bacterial mat. Really dark sediments.
Okay. Keep going. There's either a biomat or a big fish out there. Get a little closer
Understood.
and find out what we got.
Thanks, Bridge.
It's a biomat with a little fish behind it.
We're still finding them, though. There's still some activity at some point in this area.
It's been fairly consistent.
The snipe eel you're watching has a big red patch to it. And we captured one yesterday inadvertently because its hook-shaped teeth caught in one of the Velcro straps on the ROV and came up with the ROV. So we had an unanticipated collection, and we got some great photographs and discovered that it had a shrimp in its belly, which is creating that bulge and the red patch. Very translucent bodies. The jaw is needle-like until the ends, where
Ooh, we got a big crab.
each
coming.
end curls outward, upward and downward. And then the teeth are sort of like a python, where they are angled backwards to hold on to their prey.
I wonder if this is a Rachinia crab in the distance here. It looks like it. And I think we saw one of these, or a couple of these, at the very beginning of the dive, but we haven't seen anything
Yeah.
except the cancer crabs in a
That's right,
good
we
long
saw
time.
a trio.
Yeah,
We were
a trio.
wondering if they were a family or fighting each other.
Yeah.
They sure left to life.
I like how the tips of their legs are curved inward to give them a little more surface area for walking on this muddy surface.
Does he have super long claws with little baby
Yeah.
pincers?
Yes, he does. Getting closer, video. Yeah, he's playing. He's challenging me.
Such long legs, I bet he can move pretty fast.
I'm just gonna guard him here.
See if he crawls in the drawer. Take him home with us.
He just crawls right under us.
Shoot
Just
some
gonna
stills. I think he's too low for stills now, but
Too low for stills, yeah. Where are you going, guy?
He's definitely coming to check us out.
And
Or maybe live underneath. Not for long.
Is this hard substrate?
and he's gone.
Brave.
All right.
Doesn't seem to bother him much.
No, we don't. All right. See you later.
is that the direction you want to go? Or
Yeah. There's a mat here. There's a mat up there.
Okay. You want to turn right a little bit, Lars? Maybe 225. Okay.
Bridges is RV nav. Yeah, I'd like to move 20 meters, bearing 225 degrees, speed decimal 2. Good copy.
And a couple more bacterial mats coming up here.
The fish in the hole.
Might be a black belly rosefish laying in the pit, burrow, X-burrow, whatever it is.
One eye is fixed on us. Now.
Pretty astute notes over here, co-pilot. This you? Yeah. Copy that.
Not sure where that's from. It might be my running away from krill. Dust coming from behind me. Catching up. Okay, well, we saw a whole bunch of those. You can keep this zoom. We'll just mosey over the mat here.
Go ahead, Bridge.
Understood. Okay. Okay.
Come on
Understood. Yeah, if you get uncomfortable, you can stop the move and change heading too as an
So
option.
Cheryl, one thing I've been curious
Understood,
about
yes.
is you know a lot about seep environments, chemosynthetic organisms. How did that evolve from your interest in deep sea corals?
Yeah, so we were talking about the authigenic carbonates that are formed in and around seep habitats, and in the Gulf of Mexico there's a lot of those authigenic carbonates, and as the amount of venting of gases from the seafloor starts to subside, those rocks, authigenic carbonates, are often inhabited by deep sea corals. And so that was my first foray into seep habitats. And then we kind of keep finding seep habitats close to other places that we're investigating. So with the Mid-Atlantic Canyons project that ended just a few years ago, we were characterizing 2 canyons, and at the mouth of each canyon, I guess in Norfolk Canyon was a little further up into the canyon, in Baltimore it was deeper. We've found chemosynthetic habitats that were right there. So as part of characterization of other habitats, we wound up finding seeps, and now the current project that I'm working on with it's a joint agency project with USGS and NOAA OER and BOEM. We're looking at cold water coral habitats, canyon habitats, and seep habitats. And as we've been discussing during this dive, we're finding that this is actually a very active area for seeps where it wasn't thought to be for a long time. So that is now one of our habitats that we're trying to characterize where they are and how extensive they are, so that we can make sure to, or we can become better at finding where they are and where there may be sensitive habitat to avoid in the future if there are activities, anthropogenic activities occurring on the coast out here, or off the coast in the deeper waters. And it seems like human activity continues to move into deeper waters, so it's very important to determine and have ways to find where these types of habitats are, and characterize them and avoid them as we do things such as look for oil and gas,
I like them on.
drilling activities, and even placement of renewable energy, such as wind turbines off the coast, and even fisheries. All of these things could have an impact on these
Andy,
habitats. And so determining where they are is the first stage for kind of conservation and spatial planning of the use of the seafloor.
Going uphill.
Yep.
Copy.
Upslope, chasing
There's a lot of activity here.
Yeah, I find that as I see
like
more of
10% of what it was
what
earlier.
the deep sea looks
Maybe
like, of
5%.
course, my interest broadens, and you're in a
in the water.
research-oriented facility. It's great that you've been able to focus on some of these areas and really dive into them, no pun intended, but to really understand some of them.
Yeah, and again,
oh my goodness,
the partnerships and bringing so many talented scientists into the fold on these larger partnership and big initiatives
the next name of Sharknado
really
series.
Gets us far fast.
Go ahead, Bridge.
You know, besides the agencies, there's also a group of academic scientists that has been involved in all of these projects.
We'll do a quick zoom of this guy as I come
so
by
we have
just to see if there's
a
any
lot
bubbles.
of variety in scientists and what they study, and so
do you have a sense on where the current's coming from right now?
An amazing array of scientists involved in the project, and try to put all the puzzle pieces together that each of us can decipher and bring to the table, so that we get the most comprehensive characterization that we can over a short period of time.
Okay, come on out.
Yeah,
I'm
one thing that's really
to see any bubbles,
about this chat room is, of course, we have so many scientists literally from all over the world. Right now we have someone from Japan and someone from Moscow on. We've had
so we got a little time here.
Norway and Germany represented. But not only are they giving us their expertise from the research they've done or papers they've read, and they'll share with us a link to a paper, but also just their anecdotal experiences, having seen certain behavior. They may not have published on it because they didn't have video like we're collecting, maybe, but just stories that are really, they bring home the nature side of this too. They're great stories, and to compare what people have seen elsewhere is really, really important. We've had people talk about, well, you've talked about Glacier Bay and the corals you saw there compared to here, and we've had a lot of people who are involved with the Pacific expeditions on the Okeanos Explorer and the Gulf of Mexico. And,
this one's large.
We see similarities and we see a lot of differences too.
Come on in.
That's all in the nature of research, but it's also in the nature of exploration, where every new site opens up new views. And that's why we call this Windows to the Deep, which builds off of what Carolyn Ruppel's group did years ago with ocean exploration.
Earlier,
that was 2002, 2003, right?
Yeah, I don't remember the date, but so this is a follow-on to that idea of just taking peeks into how the world looks at these depths and places we've never seen remotely, like this with video.
I like that.
Viewed many of these with remote sensing techniques, so it's great to have this amazing video. and to have everybody see it at the same time and to have it archived and available to the public. And for those of you listening, you may not realize that there are teams of scientists, some of whom are on the chat room and others are dutifully typing away and annotating everything we see and listening to the comments.
Move the whole anemone.
And using a system called C-Scribe, where they can, using a timestamp, identify what they see, including what the substrate or what seafloor looks like, what kind of organism, what other organisms are there, what the conditions are like. And it all has a timestamp so that a researcher can look at the database, look at the timestamp, and then go back and look at the video for that time.
I'm surprised it's not
slice.
larger.
So
that one got away.
An amazing wealth of information.
But
one of the times that stands out for me on this expedition of the great opportunity for telepresence helping us out was when we found that black sponge, which was
Let's go see these mats over here.
Many dives ago now, but it was on a coral mound, and we were pretty close to the end of the dive, and we came upon this black mass on the surface of the coral rubble, and
like an ooze, a black ooze.
And we are all chatting in the chat room about what in the heck it could be, and we had all sorts of ideas floating around. And then I think it was Iris from the Azores who had seen this genus of sponge, and so she was able to say that it's probably Dersitis, and none of us had any experience with that, but we came from wondering what phylum it was, even to we even might know the genus, and that was amazing to me.
And we collected a sample, so it'll be verified at some point.
Mm-hmm.
Please was off.
Yeah, that was Iris Sampaio.
Mm-hmm. That's right. And we sure have had a lot of examples of the Cereanthid tube anemones catching crustaceans in their sticky tentacles. And Tina says that she's 99% sure that this is Pachycereanthus borealis that we've just observed. And she said that they actually have quite a powerful sting.
Laser's coming off.
No bubbles
Can
here.
we get a tight zoom on the little white things? I don't know if those are tubes or edges of shell material.
I think that's what we've been seeing.
It looks like the shell material almost. I can't tell. Is that what you were referring to?
Yeah, at some point somebody said that those could actually be microbes. But
so they're little tube-like microbes?
That's what somebody said.
Okay.
I don't know if I don't remember. It wasn't today.
Oh,
it was a couple of dives ago, I think. So I'm not sure, and if anybody else has information about those white tiny tubes, we'd love to hear from you.
Earlier we saw off to the side of a few of these little areas of bacterial production, we saw broken shells and looked like from bivalves. And we have yet to see any living bivalves, so we saw 2 pockets of broken shells farther down on the slope. So I'm curious as to where they were growing and where they were alive in the first place.
Mm-hmm.
almost at the end of your tether.
Got that. Oh, look at all these.
Yeah.
Those are probably out of reach for
And Nolan Barrett thinks it was Bob Carney who made that suggestion that those little white tubes were made from microbes. And it wouldn't surprise him if the microbes did form macroscopic tubes. So Tina's pointed out that the outer ring has more of spirocysts, which are the sticky cells, and then the inner ring has a lot more nematocysts, which are the stinging cells. And that makes sense considering what we've seen today.
Yep.
What's that thing in the middle of the screen?
I think it's just a
Do you want to
pit.
zoom on it?
Oh, okay.
We're seeing the dark material. Yeah, your eyes definitely play tricks. You think of something as being a mound and it's a depression instead. And certainly the size of an object plays tricks on me all the time.
Mm-hmm.
You want to come in and just check it out video, unless you can see there's nothing.
Well, we've been fooled before, but never say never.
I like that laser action there. Co-pilot.
I've been practicing.
So yeah, I guess whenever they're done with their ship heading, we can get a move in.
Do you run out of things to look at?
I'm running out.
Okay. Let me know when you run out.
I'm out.
You're up.
Yep.
I don't know if we've said it in a while, but these laser beams are 10 centimeters
Coming
apart.
back.
And we use them to help us
Okay.
measure objects because we are so easily fooled at scale.
Come out in a video. Let's see if there's anything.
All right, I'm going to put in a move for you. I'll let them stop their turn
What do you got?
at 225. I'll do 40 meters.
Oh, nice.
Delta
Okay.
Yeah.
225
Yeah.
copy.
I'm not seeing anything here. Video, are you?
Bridge,
Nope.
Nav.
Okay. Let me just pan up real
Nolan
quick.
Barrett also
You in a position
noted
where you can
that
stop the
he's
turn
done a
and
lot of
put
work,
in a ship move for
or a
us?
bit of work with marine and terrestrial microbes
40
to look
meters,
for bioactive
bearing
What is
compounds.
this? You want to come in?
25,
And they
again?
always
Speed,
surprise
decimal 2
him
knots.
with the macroscopic structures that their colonies can form,
That's
both
correct.
in liquid media and on
That was
solid
me.
media.
Yeah, I think you're still.
Okay. I'm
Nolan
actually going
is
to
at
come out.
the,
I'm going to come back.
Yeah.
Natural Products Laboratory of the Medical University of South Carolina, where they
All right, looks better.
are looking
Does it look
into
better?
some of the potential pharmaceutical properties of sponges and corals and other
All right,
organisms
so I'm lined
in
up
the
at
deep.
225.
Copy that. Coming back.
Tether pulls having me rotate though. To
Two
forward.
He
center.
hopes to
Looking
do
more
his
like
PhD
210.
on this type of work, so
Are we
very
full wet?
active contributor, and we
not quite.
appreciate your remarks, Nolan.
Yes, we do.
I can see.
That is correct.
All right, that looks much better.
Do we have a certain
Nice
destination we're trying to
mid-water
reach to?
views
Yeah, we're trying
right
to get
now.
to waypoint 6.
Tina
Ooh.
4.
But we're trying to follow the,
Is that one of the low weight kind?
following
It sure
the,
looks like it.
bacterial mats,
And a black
Copy
belly
that.
rosefish.
sort of heading in that direction.
I used to be under the impression that black belly rosefish occurred on coral mounds, and that was it.
Yeah, that's
And
it.
then this cruise has proven that extremely wrong.
Yes.
Much better.
On sedimented habitats, on rocky habitats.
Rocky ledges. Yep. I don't think we saw him in the deep waters, so at least
Nope.
still with the idea of upper, 600 meters,
Couple
something
pings.
like
Yeah.
that.
So I think when I was nav, I noticed 5.
At least I don't think we saw him in the deep waters, so at least
Nope.
still with the idea of upper, 600 meters,
Couple pings.
something
Yeah.
like that.
So I think when I was nav, I noticed 5.
At least I don't.
Mm-hmm.
Yeah, let's see what happens if you stay close to Sirius.
Stay close.
Yeah, basically try to stay under the ship, and then we'll have you push out and see if we get bad pings. Because I haven't seen any bad pings recently. How do you get rid of the trail? That's
You have to change the time and then change it back, like change it to 99 seconds.
And we are still seeing some bacterial mats. Been fairly consistent, but smaller mats as we've moved along. We're now at 391 meters. And we are about 26 miles off the coast of Pea Island, North Carolina, in the Outer Banks.
Thank you, co-pilot.
Here we
I
have
got
another
Oh, look at this guy.
glowworm.
I've already seen these, I guess.
Yep. Oh,
Darn.
oh, we've seen a
Oh,
lot of those.
it's his whole house he's moving.
yeah.
Mobile home.
And another cancer type crab.
Pilot, you're starting to go towards my port a little bit.
But I have to go see the sea star.
What's going on in the center here? Squid with
Turn if you want.
What's center? What do you see? This guy?
Oh yeah, there's the squid.
yeah, somebody was just pulling the worm.
Looks like he's struggling there, but
And pilot, I'm looking straight down.
Copy that.
There,
Somebody
try-
was
Anything
trying
for our
to swim
pinks?
with the worm.
Nothing.
looks good. Why don't you try pushing out now? Try to get away from the ship.
and we're seeing more of this really dark sediment and some bacterial mats and some pits now. Might be burrows.
What is this?
Almost look like.
Might be a very large Ceryanthid tube anemone here.
Quick zoom while we fly by it here, video.
That is very large.
Perfect.
Very tall. Has the same tentacles.
Mm-hmm.
Great. Thank you. Thank you, video.
Very
Thank
pretty.
you, Bridge. You're good to continue your rotation. Sounds good, Bridge. Oh, there you go. There's your krill.
Ooh.
You might have.
Maybe we'll
might
get
have
a whale.
Tornado going on.
You don't want to enjoy it for a moment?
Just tilt down for a second.
I would like a whale to come in now.
Go 90. It's straight down. It's a nice
Yeah,
density.
it's not.
Come in video.
We saw one of these eat a squid earlier.
One of the.
One of the fish.
Oh, cool.
I almost hesitate to zoom in on these. I'm afraid of who's going to come in and be gobbled up.
Well, that crab's attacking D2.
Definitely seen these being predators today.
I really want to
He's
see him
thinking
go
about
for
it.
one of these quill worms and figure out what he's going to do with the tube. Come on.
Looks like he's thinking about it.
We've had a suggestion for a film, just all the different feeding that we've observed. We can call it dining in the deep.
Yeah. We sure have seen a lot.
We have plenty of material for it.
Be our guest.
Yeah, I'm sure the
Doesn't look
video
like he's gonna
folks
go for it
just want to
today.
hear these ideas. They've been at it from day one, making short videos, and each one of them does a long video as well. A long story, I guess you call it.
Ooh.
It's hard to believe it's been 3 weeks. Have just flown by out here. And I've been just so happy to have had the opportunity to sit in this chair and next to Leslie and get to know you and get to know so many very unique habitats off the southeast coast here over these dives.
Yeah, it's been incredibly satisfying,
We got
exciting,
shrimp.
and so educational.
Gone.
I don't think I've learned this much in 3 weeks
Yeah.
in years since undergrad, maybe.
And, you know, the excitement of just seeing something in the field of view that you just didn't expect, and all these interactions among the organisms and chatting with the folks on shore. It's just been an incredible experience. I've been out to sea a lot, and they've always been research-oriented cruises, or I've also done education where I've taken students out to sea to train them in oceanography or seafloor mapping. And this is so different from all of that because of the telepresence
Nothing there.
side of it.
See anything
And
video?
it's been
Nope.
very exciting.
Come on out.
I can't wait to take these experiences and the video clips and the photographs that we've grabbed from the screen grabs, take them back and build curricula for my undergraduate students, and maybe even work with some teachers. So it's just been sort of all-encompassing for me. The discovery, exploration, education for me, and educating others and reaching out to the public. It's just been great.
It sure has. And another part that I didn't anticipate is the interaction through social media, and how days after we see an organism, there are still scientists trying to figure out the IDs and sending them our way. People calling the ship to give us IDs, and it's just it makes everything accelerate in how fast you can learn about these habitats and identify organisms when you have a whole cadre of people trying to help you, and it makes us leap forward instead of crawl forward.
Another
What
movie
we
if you
know.
want.
Yeah.
Good copy.
And then in a month and a half or so, I'll be back out in this area, and some of the areas that we have traveled through and done dives at, with another project that involves NOAA OER
Yeah, we're gonna do
and
this next
BOEM and
move at
USGS. and
Copy
that's
that.
the Deep
2-0
Search
copy.
program,
Bridge,
and
RV
that
nav.
stands for
Look at
Deep
these two
Sea
guys.
Exploration to Advance Research
Like to
on
request
Coral
a ship
Canyon
move,
and Seep
range
habitats.
40 meters.
And again, that
bearing
is a
220,
big
speed
multi-agency
decimal P knots.
project, and the data that we've collected on this cruise is going to help us so much
That's correct,
when we're
Bridge.
trying to determine where to go and dive, and what kind of
No, you have one
habitat
open,
we
one
might
closed.
find, and even the mapping data that this cruise
come in video. Let's take a look at
has
this.
collected. Every night we've been mapping parts of the seafloor, and all of that information will help our program, the Deep Search program, going
Kevin, a little bit
forward,
more,
and as
if
well
I
as many other
look at this
scientists.
empty guy.
And the Aspire
What is all
program
that
is
around
another
him?
one, and that's the Atlantic Seafloor Partnership for Integrated Research and Exploration, and that's
That's
kicking off
little
with
worms.
this
Those
campaign,
like pieces of the anemone.
and it's led by NOAA OER, and that has we're trying to look at connectivity across the
Okay,
whole
you come full
Atlantic basin in this Aspire campaign. So all of this video and everything else that we're collecting will help move that project and initiative forward as well.
Take a look at this, Matt.
I'm sure you saw the remaining tentacles on the seafloor of that tube anemone.
I was wondering what that was.
It looked like it.
Yeah.
I'm waiting to hear back.
See anything?
Something must eat the tube worms, is what Tina suggests. I agree.
Video? Come wide, video.
And pilot, yeah, we crossed over one of those large shrimp. If he might be out of view, I was hoping to capture him.
Where is this?
Well, he's now under us. I didn't know how far away
Oh.
we still were.
We might see another, hopefully.
I will keep eyes out for.
Okay.
I think we saw a couple of them, so
Okay,
pretty sure
good.
there'll be
Yeah,
some
it'd be
more.
good to get another good view of them.
Copy that.
Some more mat up there.
Yep.
Yeah, maybe that crab is the culprit for
What
the
is
anemone.
Oh, that's a squid. Okay.
Yeah, Andy, you should go back and look at the beginning of the dive after this.
Yeah.
I didn't know that nudibranchs were one of the predators on cereanthids, the tube anemones. Cereanthidivores. I've never heard that term before. Dendronotus and maybe other nudibranchs.
And nudibranchs are shellless gastropods, correct?
Mollusks, yep.
Mollusks. Yeah, they're not gastropods. They're
No.
a different class.
Right there.
Just a quick zoom here, video. What is this? Oh, it is. It's a buried
It's a mud star.
mud star.
Those are one of the few echinoderms we've seen on this dive today, aren't they?
It might be the only one, actually. I mean, we've seen several of this.
Right, yeah.
But,
The only species.
Yeah, I can't think of anything else. And I'm sure Nolan's happy to see it. He missed the earlier ones. It's possibly a Plutonaster.
Okay. When you're. Does it move? Oh, it's burying itself, isn't it?
We've seen them bury it. He's doing it really slowly if he is.
Look at this. Zoom in right here, video. Maybe he's just slithering around.
Yeah, when they're ready to come out, they lift a leg and knock the sediment off of each of the, I should call it an arm, each of the rays. I think he's just flexing his knee.
Okay, come on out, video.
A Plutonaster.
So that is the same species we've seen in the past.
Okay, looks like a pogo stick.
Another Barracuda.
Full wide video.
Cancer crab.
Going back to the nudibranchs that we were talking about that eat cereanthids, they are opistobranchs in the gastropod group, and apparently they're quite lovely to look at. Maybe we'll get to see one, or I'll look it up after this.
Quick zoom video.
And
See if
they
you
actually
see
can
anything.
steal the nematocysts and become stingy themselves. Well, that's pretty amazing.
Nope. Alright, come wide video.
Go ahead. Yeah, one second, please.
Another star.
There's like two of them here, I
There's
think.
one of the mud
Yeah.
stars,
One in the back, too.
self-excavated.
Yep.
Come on in video. We can do a quick zoom on it. Just get a little closer on it. Yeah.
You can see his pointed tube feet.
Looks like he lost an arm.
Yeah.
They have, oh, you can see one of his
Looks like
rays
we
has
have
been
just over an hour.
damaged, and he is regrowing that one in the upper left.
Yeah, I'll lay in there for you, even though.
And it looks like we have just over an hour left on the seafloor today.
All right, pilot. I'm looking right down at you.
Alright, come on out, video.
90 degrees.
Let's push ahead.
Yeah, you have time to rotate. Good copy. All right, that move is done. They're going to rotate their head around.
You got a nav.
You were looking for the larger shrimp than this, right? Watch lead. Oh, I was asking about that shrimp. I think you were looking for the larger version.
Right at just off screen on the bottom.
What?
We just went over one off screen on the bottom.
Oh, okay. Yeah, let me
He
know
was
if you see
buried.
another one.
Yeah, he was buried.
This
I
is
had unkeyed from
probably
you.
the longest stretch we've had with so few of the bacterial mounds.
There's one on
Yeah,
the
there's
left.
one there,
To the
but we
left.
just went through where there were none in view.
Yeah.
We have had very little of that today, and there's another view with
Oh, that's neat.
another of those tube anemones. They're quite large here, aren't they?
I think I see a mat up there, too.
Yeah, these are very large.
Is that one of the shrimps video? Or is that something?
No.
Okay.
The ones we have would be 10 centimeters,
About this.
extremely long antennas and
The
partially
water
buried.
temperature down here is about
Let's do a quick zoom
8.4
here.
degrees C. So when we bring up a sample
Like that?
Nope.
Oh, that's a
in our
different
It's a
bio
shrimp.
different
box,
one.
it is
Ah.
a cold dip for
Yeah.
our hands to go collect it. Especially when I have to scrape up the sediments from the bottom. Hand pretty much goes numb after a few seconds.
And that looks like a different shrimp than the one that we've seen buried, partially buried.
Oh, maybe 5, 7 centimeters.
Yeah, something like 5 centimeters long.
You can go full in video if you'd like. Just to.
Yeah, as long antennae are red instead of the long white ones that we've seen.
His legs are so delicate.
He's cleaning them off.
Good video. Yeah, he might be. Maybe
It's
it's just
funny.
talking. Not sure.
It might be. Come on, have video.
It's a great shot.
All right. Third type of shrimp. Whoa, what is going on over there?
Looks like a barracuda has
Fish
hit the
dive
sediment hard.
It's made landfall.
Yeah. Messing up my shot.
And we've seen a lot of these midwater organisms just
Hey,
kind
Josh,
of
have you flown today yet?
hit the sediment really hard, and basically people
What's
have
going
told
on
us
here?
that
Quick zoom here video.
that's a
It's a snail.
Oh, that's a big trail.
Yeah, I grabbed one of these earlier today.
Come on out, video.
that they get when they get nervous and confused, they try to swim down, and they probably can't see because of the lights, and they swim right into the sedimented surface of the seafloor here.
Yeah, I flew from like 9:30 till 11, and then Fernando flew from 11 to 12.
All right. Gonna take the whole pilot shift then.
Has Sean flown?
Yeah, he said he did.
Oh, he did launch. No?
No, I said nav. Deck.
Like I said, whole pilot shift.
Yeah, take the whole
Couple
thing
of
in.
black belly
I'm just kidding,
rosefish.
Sean. You want to split it?
Nah, it's a short one. Can I have it?
What was that? Watch leads? Did you say?
They were just commenting on
Oh.
It's a couple more pings of D2.
Whenever I lift a little bit off the bottom, it starts to go crazy.
That's interesting.
I wonder if it's repeatable. Let me stay real close to the bottom.
You said you think it's when you were off bottom?
I was like 2-3 meters off
Huh.
for a second. But maybe it's getting like a weird reflection or something.
Like a weird bounce or something? Yeah, I don't know.
Good copy. You guys ready for a move?
Yep.
Yeah.
Definitely.
Where
We're
are all
gonna
the
do,
seeps?
220 again.
I'm not sure if our
220.
last waypoints were
Could probably will allow it.
sites that had been identified, or if we were just going to keep climbing to see if we could find any that hadn't yet been identified from sonar.
Bridge RV
I
nav.
don't recall.
We're good for a ship move here.
All right, check it out. I'm gonna go. I'm
40
gonna go up a
meters,
little bit and see what
bearing
happens on the
220,
scale.
speed decimal 2
Yeah,
knots.
I'll keep track of high pack and
I
altitude.
think that we were the last move was
That's
just
correct.
continuing up the slope,
think
but
so,
didn't
too.
have
Yeah.
target, a seep target.
Roger that.
Okay, come back down.
Coming down.
We started this dive at about 520 meters, and from the very start we began to see evidence of seeps. Saw patches of those bacterial mats that right now we aren't seeing any in the field
You
of
want to
view.
try changing your
But
heading?
if you've been watching at all, you've seen several of them pass by
To what?
throughout the dive. We've seen them.
Try a different heading. see
And
if you get
somewhere around
bad
the
things.
400 meter mark, we began to see
Oh.
more and more of them. And at one point, on every horizon, it was just a sea of these
little
bacterial
pivots.
mats. And we did see bubbles emerging from beneath the mats in
Yeah,
several different places.
Doing the service and go around filling them
But
up.
clearly an active area.
Create a nice
These
smooth
obvious
surface.
bubble plumes are known to come and go, turn on and off. We've seen that here. We've seen it in many different places where seeps have been studied and seen firsthand, either using submersibles or ROVs. And also through the remotely sensed data, you can pass over a known seep site on one day and not see bubbles, and the next day you do see bubbles. We did that little experiment off the
that was a
coast
ping?
of Oregon,
We're back.
and
So
I didn't do anything.
colleague
looking in line with
Brendan
the ship.
Phillip did a lot of work with that for his
Oh, it was.
doctorate
Okay.
I don't know if that
at University of Washington.
was just a single ping. You could rotate 90 and then try to do it again.
Okay.
We tried to measure the bubbles to see how they grew from ground up. There were a couple of
And you're
students
just
on board
off
from
to my
University of
front
Washington, graduate students who were trying to figure out a way to have a scale in the image. I don't think the results were really what they had hoped in terms of just were they even able to measure it. I don't think they ever took that data any further, but I could be wrong. I left at that point. But I know it was an exercise in frustration, trying to measure bubbles as they arise through the water column. So if you're viewing now and you wish you had viewed earlier to see the seeps, these the dive video are replayed every evening. And so the highest concentrations that we saw of the bacterial matting was probably in the first 2 hours, certainly 2 to 3 hours. But we are seeing fewer and fewer of those patches.
Could we take a look at the one of these fish? Not the one that's hitting the bottom, but the ones that are laying on the seafloor?
Sure, like the one to the left here?
Yeah, thanks.
Absolutely.
Wondering whether we have a good ID on this or not.
Sediment picked up right here, too.
Yeah, it sure did.
Where are you going?
He wasn't.
Oh, we're going to get him.